There's Emma.
I just promoted her to panelist.
Sweet.
All
I
right.
wasn't sure if you could.
She says she's rejoining the webinar as a panelist. Great.
Doug wins. Got her first.
Yeah.
Quick trigger finger.
Good job,
Awesome.
Doug.
Good evening, everybody who's joining us. We'll get started in a few minutes. Welcome, Emma, as another presenter. And we are recording. All right. Good evening, everybody. Welcome to tonight's seminar on environmental DNA as part of our Earth Day Every Day webinar series. I'm Doug Zemeckis, an extension agent with Rutgers University Cooperative Extension. I'll be your host this evening. We have three guest speakers, Jason, Aaron, and Emma from Monmouth University presenting. Tonight's seminar is number 2 out of 6 in this mini series in the spring. The elements of natural resource management. We started off last week with a talk on forest fire management and forest ecology. And next week we have a speaker on Dr. Joe Paulin on black bears in New Jersey. This program is part of our education outreach through Rutgers University Cooperative Extension. It's a collaboration between 2 outreach programs we have, Earth Day Every Day, and this is the Earth Day week, obviously on Monday night. Here we are a few days later, and also the Marine Extension Program Seminar Series. So we have a team of extension agents from Rutgers Cooperative Extension collaborating to bring, organize, and help orchestrate this educational outreach seminar series. And we cover a variety of terrestrial and marine related topics. Tonight is a marine topic with environmental DNA. Some navigational aids for tonight's webinar. We're scheduled for about 1 hour, including a presentation and questions and answers at the end. I'll be moderating the Q&A at the end. When you have questions for the speaker, please type them in using the Q&A function, and then we'll get through as many as we can at the end of the presentation. Next slide, please, Jason. At the end of the presentation, one of my colleagues will launch a poll, which is a survey to get your feedback on what you thought of tonight's seminar and what you learned during tonight's presentation. Our Institutional Review Board, IRB, at Rutgers University views that survey as a research project that we use to help improve the quality of our education and outreach projects. And you will get asked for your consent at the end and right before you choose whether or not to take the poll in order to participate in that study. We appreciate your valuable input so we can improve our programs. Next slide, please, Jason. And Rutgers Cooperative Extension and our collaborations with multiple counties, state, and federal levels, including at the federal level, the U.S. Department of Agriculture, make acknowledgement that us at Rutgers Cooperative Extension, we are an equal opportunity program provider and employer, including programs such as this tonight with our Earth Day Every Day outreach seminar series, that we provide these and other programs without discrimination to as many people as possible throughout New Jersey and in a variety of formats to facilitate participation, live, and also recording to tonight's webinar to make it available to others afterwards. Next slide, please, Jason. With that, setting our sights on tonight's seminar on environmental DNA, or eDNA, we have 3 speakers joining us to cover their work on these topics. From Monmouth University, we have Dr. Jason Adolph. He's an endowed associate professor of marine science at Monmouth University. He specializes in phytoplankton or algae ecology, harmful algal blooms, and real-time water quality monitoring of the coastal ocean. And a lot of his time is also spent on eDNA that we're going to be learning about tonight. Joining him is Erin Conlon, who's the community science coordinator at Monmouth University as a part of the Urban Coast Institute. And she spends a lot of time working on the community science side of eDNA and other water quality monitoring projects. And the third presenter joining them tonight, we have Emma Najarian. She's a high school intern volunteer working with Monmouth University on some of these projects, joining us to share some of her components of this work. So thank you very much to all 3 of you for joining us tonight. I will turn the controls over to you to teach us about environmental DNA.
Thank you very much, Doug. And let me say that we're all excited to be here and grateful to have some time to share with you this work that we've been doing in environmental DNA and fisheries monitoring for offshore wind. And like Doug said, my background is in phytoplankton ecology, and I really got into the fisheries world through this technology called environmental DNA, because from the mechanistic side of things, how you sample it and how you interpret it, it has some similarities to how we phytoplankton ecologists operate. So the story, of course, as a lot of environmental stories do, starts with climate change. And in this case, the impact that climate change is having and is predicted to have on fish. And I found this infographic on the web that I think does a good job of describing things we probably all know: where climate change comes from, loading of the atmosphere with heat trapping carbon dioxide and other greenhouse gases, primarily from the burning of fossil fuels. What I find a lot of people don't know is the impact that this has on the ocean that we don't see as land-dwelling animals. About 90% of the heat trapped in the atmosphere goes into the ocean, and we don't feel it. And that's also true for carbon dioxide in the atmosphere. And both the heat and the carbon dioxide have impacts on the ocean's physics, chemistry, and biology. In regards to fish, one of the things that we see happening is movements, changes in movements, changing in migration timing, changing in range distributions. Southern species are being found further north, and shifts of that type. So we as people living on the planet are trying to come up with plans to mitigate climate change. And one of those plans is to shift away from energy sources that put carbon dioxide into the atmosphere and move towards more such called renewable energy sources. And offshore wind is one of those energy sources that a lot of people have invested a lot of time, thought, and money into as a potential solution to moving away from fossil fuels. The maps you're looking at here are the East Coast and the West Coast of the United States, and I'm focusing on the East Coast. And each of these colorful polygons we're looking at is an offshore wind development site that is planned to be loaded up with wind turbines that will capture wind and produce electricity through a generator attached to the turbine, the same way that generators at hydroelectric and coal or oil powered electric plants produce electricity. And you can see there's a lot of offshore wind planned for the eastern seaboard on the continental shelf. We have good winds, we have shallow water, we have a good environment for doing this, and lots of different companies are planning to do so, as is plainly evident from this map. On the West Coast, where the water is deeper, while our pylons will be attached to the sea floor on the continental shelf, which is 100, maybe 100, 120 feet deep, these turbines will be floating in much deeper water, and they're a little bit behind us in terms of schedule. Now, the plan for New Jersey is 100% clean energy by the year 2050, and Governor Phil Murphy has issued an order that he wants to see 7,500 megawatts of offshore wind energy by 2035. And recently, in the recent solicitation award, a little more than half of that has been awarded to two different companies. So it's moving forward, but it's important to note, before I leave this slide, that nothing is being built yet in these areas. We're still in what we call the before construction phase. So far. Now, New Jersey has a program called the RMI program. It stands for the Resource and Monitoring Initiative. And this is a program that's set up that the wind companies pay a fee, so to speak, to the state to support monitoring of natural resources and the environment for potential impacts of development of offshore wind. And what I think is special about this program compared to working directly with wind companies, which is another thing that a lot of people, including ourselves, do, is that although the money comes from the wind companies, it's being administered through the state. And so they take a more, they tend to take a more regional approach to asking questions about the impacts of offshore wind on the marine environment and natural resources. In other words, they're not tied to one wind site or another and the adjacent areas right there. They're more interested in the coastal resources as impacts New Jersey as a whole. And so I find that they have I like their regional approach to how they approach monitoring for potential impacts of offshore wind. And the RMI program is a program that funds much of the work that we're going to talk about this evening. So any company that is planning to build a wind farm, and they're shown again here, has to show that they're monitoring the environment and monitoring the marine life in the area where they're building. And a lot of times that comes down to what's called a fisheries monitoring plan or an FMP. And here, we at Monmouth University have been fortunate to collaborate with Doug and others at Rutgers University on these fisheries monitoring plans because they're huge ordeals. They take a lot of time and effort to do. And what a fisheries monitoring plan in simplest sense asks is what fish are in the water where the wind farm is planned to be built and around where the wind farm is planned to be built, and what effects might that construction process and the installation of the wind farm have on the fish that are there. Traditionally, fishery scientists will look towards their traditional toolkit, things like I heard described recently as trawls, traps, and hooks to ask those questions. Capture surveys. You go out, you trawl, you trap, and you fish, and you find out what fish are there, and you do that over and over again before, during, and after. And what we're doing is a little bit different from that. We're using a technology based on environmental DNA, which is non-extractive. When we go out, we're collecting about 1 liter of water, which is about half of a big bottle of soda. And within that water, we're finding the genes that tell us what fish have been there within a very recent time frame, within a couple of days. So I'm going to turn this over now to our high school intern, Emma Najarian, and she's going to explain in some more detail what environmental DNA is. Emma?
So the idea that comes with all this is, what is eDNA? So eDNA, or environmental DNA, is made up of any DNA in the environment that has separated from the original organism it came from. This includes things like blood, mucus, scales, tissue, poop, and some other waste products that come from organisms. And like Dr. Adolph mentioned, the DNA that we find is representative of where a fish was within a few days of collection. So we use eDNA in a similar capacity as they do in forensic crime scenes, except instead of looking for criminals, we're looking for fish. So we collect eDNA in those 1 liter ocean samples, both offshore and on the shoreline. Then we take these bottles of ocean water and filter it, collecting DNA on a filter. And then from those filters, we extract the DNA through a bit of a lengthy extraction process and end up with a small tube filled with DNA. You can switch the slide, Dr.
Hmm?
Switch the slide, please.
Gotcha.
Thank you. So then next, this tube of eDNA could be processed in one of 2 ways. We have option A or option B. So usually with option A, we use primers that target one specific organism's DNA in the sample. So we're looking for something specific, such as sharks, bony fish, or some other species. But then in option B, we use universal primers that look at all the species in the sample rather than just specific ones. The DNA is then amplified and sequenced using metabar coding, and those DNA sequences are coded then compared to the sequences in the reference database that we have to determine what species are present in that sample. We mainly use option B to determine fish community composition for our samples. We're not just zooming in into one species, we're looking at everything that's in that sample. Can you switch to that again, please? So then what metabar coding is, Dr. Adolph came up with this great graphic we have here. You can think of eDNA metabar coding as the barcode on like an apple at a grocery store. We use that code to quote unquote check out the fish present at the time of sampling when compared to the reference database.
Thank you, Emma. And I didn't completely come up with this. I asked ChatGPT to draw me a picture that captured the reason we call it metabarcoding for marine life and for other life is because within all of our genomes, there's a sequence that's part of it that's like a barcode at a grocery store, and it would tell somebody who's looking what species you are, and even down in some cases to what individual you are. Thank you, Emma. And Emma has worked in our lab as a volunteer since last summer, doing everything from filtering samples, going out to the field and sampling, extracting DNA, running PCR, and checking gels, and has been a great help. Thank you. So this is a new technique, environmental DNA for fisheries. It's not a new technique overall. It's been used in forensics studies and other microbial ecology for a long time. But it's a totally fair question to ask. And how well does it work compared to, say, a trawl survey? And one of our lines of research that we have done and are continuing to do is simply going out on these fish trawling missions. And this is an example from the New Jersey Ocean Trawl survey that we worked with. And as they're going through their trawling work, we ask them, collect us a liter of water and freeze it. And we're going to extract it and look at the genes, and we'll compare what fish you find with what fish we find. And to summarize the results from this particular study that we did a long time ago, about 75% of the taxa that are found in common between the trawl and the eDNA. And those are the taxa that are in common. Often we find in one sample more taxa in eDNA samples than a trawl catches. And this is related to the fact that fish can swim and they can avoid a net, and they're patchy, but their DNA is kind of dispersed throughout the water column, so it's easier to capture in a given liter of water than in a net tow. This graph here is showing, if you look at the relative abundance, so the fraction that these fish comprise of all the fish versus these fish, the fraction of their biomass versus the fraction of their eDNA reads that we find, is there a relationship? And what we found here, and what we've continued to found, is that the eDNA approach does a pretty good job at returning the relative abundance of fish, how enriched or depleted that fish is within the community of fish there. What eDNA does not do a great job at currently, and more research is needed to improve this, but trawls can do this, is the absolute abundance of fish. How many fish are there per square meter of ocean floor in a trawl, for instance? The technology is not quite there yet. So in our RMI project, we have two broad objectives. One is to create an ecological and a methodological baseline that will help us identify potential impacts of offshore wind development on surrounding marine fish communities and the fisheries that rely on them, and then to further refine the use of environmental DNA for fisheries monitoring programs for offshore wind development. Why is this important? Why is this important, and why are we doing this work? There are a few reasons that will come out in the rest of the talk we're giving. One is that as wind farms get developed and pylons are installed in higher density, it's uncertain how easily traditional trawling vessels will be able to navigate those areas and continue trawling the way they do now. So we're taking the time now before that happens to kind of create that baseline of eDNA measurements and compare it with trawling and other capture methods to see if we can fill some of those gaps that might be created by our inability to trawl later on. And use of eDNA also gives us access to environments that aren't accessible to trawl ships because of bathymetry, such as structures on the bottom where certain types of fish live, and near shore environments like the surf zone that Erin is going to talk about in a little bit. The map on the right here shows where we sample in the ocean and along the shore for this project. This project is broken up into 7 different campaigns of sampling. The first 2 here are the blue dots and the red dots respectively. The blue dots are sampled by boat 4 times a year, and they're meant to capture the composition and the variability in fish communities that are out in the ocean, in and around the areas where the wind farms are going to be built. And at the time we wrote this, these were the main wind farms that were heading for construction: Atlantic Shores and Orsted Ocean Wind 1, which, as you probably know, Ocean Wind 1 is no longer in the mix as a developing wind site. The red places are places along the shoreline where we have our team members, Erin specifically, and community members sampling eDNA fish composition from the beaches. And we included that because although the wind farms are out here, we know that the recreational fishery along the shoreline is super important. And were development of this area to impact this area, which is possible, we have to include that possibility. We want to have the before construction data to be able to get at that. The next 3 campaigns are where we're pairing eDNA with ongoing capture surveys run by the state. This is the New Jersey Ocean Trawl, which happens in this area here along the coast between 0 and 30 meters depth. The Raritan Inventory Project, which occurs up here, trawling in this estuary. And this is important to offshore wind because some of the cable routes are going to run in this area here. And again, if there's going to be an impact on what's happening here, we want to know about that. And the New Jersey Artificial Reef Survey, which occurs at 2 sites, Little Egg and Seagirt. This is important for offshore wind because a lot of people look at the installation of turbines as basically artificial reefs being formed on otherwise nearly featureless continental shelf area. So we could learn something about the fish that live on our current artificial reefs that might teach us something about the fish that will occupy the areas that are going to be constructed. And then something we're not talking about at all tonight, an acoustic telemetry project where we're using acoustic telemetry receivers. This is run by our colleague Keith Dunton. And we'll be looking at the relationship between what he detects by acoustic telemetry and what we detect by eDNA. So I'm going to turn this over to Erin now, and she's going to focus on this community sampling of the New Jersey surf zone that's part of our project here. And I just want to kick Erin off by saying the reason we included this is because we value community sampling. We have other community sampling programs at Monmouth University, and there are lots of other community sampling programs throughout the world. And we think that the eDNA approach is a way, it's a gateway for community members to get involved in fisheries sampling, being that most community members don't have a trawl ship at their disposal. But you can certainly collect a bottle of water and send it to us and participate that way. So I'm going to turn it over to our community science coordinator, Erin Conlon, now.
Hello. So you've touched on a couple of these objectives that we have for our community sampling campaign, but I'll just reiterate some things. So one of our objectives is to collect data to establish baseline conditions in surf zones adjacent to where all of these offshore wind farms are going to be developed. It's important because what's going on out there can impact what's happening on the shoreline, also where the cable landings are going. That could also impact anything that's going on there for the fishermen, people who are just in the area who want to utilize it. We also want to establish the relationship to offshore sites. So we want to be able to compare what fish composition is on the shoreline compared to what's offshore before all of the construction starts, so we can see if it changes while it's being constructed and after that. We want to compare the techniques that we have for community eDNA collection to lab-based eDNA collection, which I'll get more into in a couple of slides. We also want to engage the community. Through other programs we have at Monmouth, we found that there's really nothing more important than connecting to the communities that are around these areas, all the stakeholders that are like around all of these places every day. Because they know what's going on in their areas better than we ever could. Also, they're concerned, and especially with offshore wind, there's a lot of misinformation and questions. So if we open up that connection between the researchers and the stakeholders, typically things go a little more smoothly, and they at least get the hands-on experience so they can see what we're doing and get a better understanding of that. Go to the next slide. So this is what our kits look like. It's pretty simple. There's not too many things in there. I see the sampler seem to fall behind our bucket somehow. But that piece is the most important part here. That's what's going to filter our water. So for lab-based, we take a bottle of water and we freeze it and then we filter it. They will go out to a site, they'll get a bucket of water, and they'll use that sampler to filter the water on site. So the thing next to that is the filter. That's what they use. And it's self-preserving, so they just collect the water and they'll be able to send it to us. No freezing or anything required. We also have the X-Tech that'll measure salinity and temperature. They get a standard with that, but I'll go over that on the next slide. They get the bucket to collect their water, and they get some data sheets too. The only thing we don't supply is waders and seasonal appropriate gear, which usually isn't a problem. Most of our volunteers are fishermen or a part of a fishing club, so they already have that. You can go to the next slide. So this is our procedure for our community scientists. So the day before, they're going to calibrate that salinity and temperature pen. So they get a standard, they get step-by-step instructions, and I'm always available via email or text if they have any questions. They also want to check the battery life on their battery just to make sure when they get to the beach, they can actually filter the water. So when it's time to sample, they're going to sample alongside me. So I'm going to set a date and they'll have a week's time frame. So 3 days before the day I'm sampling, 3 days after. So that's their sampling window. So they'll go out. That's also paired with when we have people offshore taking samples. So that's 3 days they're out there. So we'll do that at the same time, just so we have similar data at the same time for 4 times a year. It's once per season. So they're going to arrive on station. They're going to go out into the water as far as they're comfortable. Depends on the tides, how far they'll go out. And they'll get a bucket of water. They'll bring it back. They're going to write their names on the data sheet, the time, the location they're sampling, their coordinates, and atmospheric conditions. So some things they measure: what's the sky doing? Is it cloudy? Did it rain? What's the air temperature, wind strength, and wind direction? They're also going to take ocean conditions. So that's just going to be tidal stage and how rough the waves are that day. So they get their bucket, they read the salinity and temperature. They're going to note that on the data sheet. Then they're going to filter. They're aiming for a liter of water. On the samplers they have, there's a bottle and it says how much they filtered. So they'll note how much exactly they filtered on the data sheet too. When they get back to their house, they're going to rinse everything with fresh water, and then they're going to send me the filter and the data sheet in a prepaid shipping envelope back to Monmouth, and then we're going to process that. Which is on the next slide. So this is just comparing community-based methodology to lab-based. So community, they filter in the field, and then lab, we take a bottle, we take it back, we freeze it, and then we thaw it and filter it. It usually takes about 24 hours to thaw the water. And then that's when the process becomes the same. So we'll extract the DNA from both of the filters. We will run PCR, so that's going to amplify all the DNA that we want to see. And we're going to send it out for sequencing, which Emma explained earlier. And then with that, I will leave it. I'll send it back to Adolf, so he will do the results that we get from that sequencing.
Thank you, Aaron. Yeah, this aspect of the project was very important to me, the community aspect. And as Aaron said, science, we want data in science, and this little difference in the two protocols for collecting and preserving the eDNA from the water before everything converges and is done the same from the lab forward. In order to expand eDNA sampling and community participation in fisheries monitoring, we felt it was really important to establish, again, a baseline for and collect data that shows us that data collected through the community method is comparable. Oh, sorry, the community member is comparable to that collected through the laboratory method. And we don't have that data to show you tonight, but what we're going to move into next is I'm going to give you a look at some of the data that we have been getting back from our program, focusing on our first sampling that we did in this program, which was in November of 2023. And what you're looking at on the map here, of course, this is the coast of New Jersey. The triangles, 1, 2, 3, 4, 5, are our surf zone sampling sites that Aaron and Emma sampled using laboratory methods. And then the circles represent places where the boat sampled. The red outlines here are the wind areas, Atlantic shores, and the wind area formerly known as Ocean Wind 1. It might still be called Ocean Wind 1. And this map is the color of the dots or the triangles is keyed for something called species richness. And this is simply a count of how many different fish taxa were detected in the sample taken at that station. And you can see the scale over here from low numbers in the blue to higher numbers in the yellow. And I want to take a second to note here that fish genes are not fish. And we have to remember that. They are from fish, and because environmental DNA lasts for about 48 hours in the environment, if we find a gene from a fish, we know the fish was nearby that area, and depending on the currents, which is another collaboration we're doing with Rutgers to look at, depending on the currents, that water that you're detecting could have moved in that 24 to 48 hours from typically about 10 kilometers distance away from where you collected it. So you can see here the difference in species richness among stations. Some stations detect a lot of taxa, and most stations were detecting around 10 to 15 taxa in a given sample. Well, the other thing I need to mention here is that these are only teleost or bony fishes. These data do not include elasmobranchs, which would be our sharks and rays. That's in a different data set that we don't have completely worked up yet. So I want to show you some examples. If you've ever been fishing in New Jersey, the damn sea robin, you know the sea robin are out there. Excuse my language, sorry, Doug. You know the sea robin out there, you know they're abundant on trawl ships, sea robins, and Doug, you can back me up on this. Sea robins are very, very common, abundant, one of the things you catch a lot of out there, and that's also reflected in the eDNA. Their DNA is present at all of the shore stations and at many, many of the offshore stations as well, where they're found. They're where we sampled. Oh, by the way, the small black dots are stations that we sampled and did not detect whatever species I'm showing up here. Other, what I'm calling widespread fish, include things like striped bass. And at this time, in November, the fall, lots of detections along the shoreline, except not up in Sandy Hook, and relatively frequent detections. The numbers over here, by the way, are the proportion of all the fish DNA that was represented by that taxon. So in these samples, about 50% of all the DNA was striped bass DNA here and a little lower here. And I promised I would tell the joke. I hope you're fishing here that day in November, not here, where there was no striped bass. I would have been here. And everyone else I know in the world with a fishing pole would have been here, of course. That's the way it always works. Summer flounder is another what I would call widespread fish. We find it both along the shoreline in the shoreline samples and in the offshore samples as well. Again, another hot spot for them here, where they at least represent a lot of the DNA. And as I showed in that previous graph, when we find a species representing a high proportion of the DNA, that usually means they also represent a high proportion of what would be a trawl capture. Black sea bass is an interesting species in the context of offshore wind because they're structure associated, and they're one of the things that are expected to really kind of like hanging out in the constructed wind farms. And so it's really important to get good baseline data on them. They would, I would say, fall into the widespread fish category, detected at 2 of 5 shoreline stations and a good portion out in the water, although their proportion is relatively small. And scup would be another one that is common in trawl surveys, and also was found at both the shoreline and out in the water. There are other fish that were largely absent from the shoreline samples, but were observed in the boat samples, and these included, among other fish, the American conger eel. This is a fish that spawned in the Sargasso Sea, which would be out kind of in this direction out here, and that then makes an eastward migration, a coastward migration, and looks like we picked up a signal from a few of them in this region here that were probably this time of year heading coastward, but not a very high proportion, less than 10% of the total DNA fish that was out there. And then butterfish also. Interestingly, we don't find them at shoreline stations except down here in Wildwood, but we do find them out in the boat. Now, I said previously that these are teleost fish, and I also said that fish genes are not fish. That's very important. They're different. And one of the things that's different about eDNA as a fish capture technique is that what we're actually looking for are vertebrate genes. We just happen to have a reference database that's filled with and targeted more towards teleost or elasmobranch sharks and rays sorts of fishes. But we also collect marine mammal data just incidentally as we're doing this. Now, if you're conducting a trawl survey, the last thing you want to see is a dolphin or a whale come up in your trawl. It's not good. They're protected, and it leads to a lot of paperwork, and no one wants to harm these beautiful creatures. But remember, this is the detection of bottlenose dolphins that are being made in a liter of water that's taken from the ocean. So these, again, we did not find along the shore, although we did find some near shore populations and some higher abundance, relative abundance populations out in the offshore regions. But we don't detect a lot of them, and that's especially true for humpback whales. So we can detect dolphins and whales, but we tend not to detect dolphin and whale DNA very much because these are big organisms and they don't shed DNA in the same way that fish shed DNA. Through mucus, especially, and just having a small body size and being very abundant. So, were we to target humpback whales, we would choose a different approach to looking for their DNA, and there are lots of people working on that sort of research. But to me, it's interesting to think that, you know, as a fisheries biologist looking at fish, then all of a sudden you could think more broadly about fish and include in the community things like bottlenose dolphin and humpback whales, which are part of the community out there. They're part of the marine community out there. And they're also, of course, part of what might be impacted potentially from offshore wind development. So let me wrap up here and conclude saying that we're employing this technology, environmental DNA, something stolen from the books of forensic science, as a non-extractive and non-lethal way to survey what fish are out there and what is the biodiversity and what is the community composition of marine life. And we're specifically trying to gear our studies towards collecting the baseline data and developing the baseline methodologies to allow us to detect whether or not the development of offshore wind in our coastal waters will have an impact on marine life, such as teleos fish, elasmobranch fish, and to some extent, marine mammals and turtles, which we can also detect, but again, we don't see them a lot. Nobody's suggesting, I am not suggesting, I shouldn't say nobody. I'm not suggesting that environmental DNA techniques are going to replace trawl surveys. I am suggesting that trawl surveys are a standard, but they're not perfect, and they can use some improvement. And I think there's room for improvement that environmental DNA can bring to how we survey fish using trawls. Specifically, we can sample more, we can sample cheaper, and in some cases, we can sample with greater sensitivity than trawls can, and with greater access to developed environments. eDNA can be scaled. We can do a lot more eDNA than we can do trawling. And if you're a marine scientist out in the audience, or if you're not, I'll tell you, one of the major problems in marine science and marine biology is collecting enough data to make statistically rigorous conclusions about what you're looking at. And that's a problem because the ocean is big and the ocean is deep and you can't see stuff in it. And then finally, as Erin went over, one of the exciting things about using eDNA in a fisheries monitoring context is its ease of deployment and the ability to offer community members a chance to participate in fisheries monitoring programs in a new way. Of course, I know Doug runs other community-based fisheries monitoring programs, and I know they exist. And eDNA just offers a new way to get people involved in a new technology and way to look at fish. And we're very excited about that. In fact, there's a picture of Emma in the surf zone. We're still looking for volunteers for some of our sites, specifically Atlantic City and Wildwood. And if you're interested, you know who to contact after the seminar. It's Erin Conlon at this email address, econlon@monmouth.edu. And with that, on behalf of Erin and Emma, I would like to thank you all for your attention and ask if there are any questions.
Awesome. Thank you very much, Jason, Erin, and Emma, for joining us tonight, delivering an excellent presentation, very well orchestrated amongst the three of you, really valuable, and covered a lot of ground or ocean floor as well. Really useful. And we have quite a bit of interest. There are several questions in the chat. I'll relay them to you now. A couple people asked, and then your last slide came, Jason. But if there are any other thoughts on how people can actually get involved, are there other opportunities on the horizon in addition to the ones you just mentioned? Can you follow up, Erin, with a little bit more?
What I can say is we are looking for those two sites to be covered. At the moment, we have the funding for those two sites only. And I think Jason might comment farther on that if we have anything planned. I don't know.
I'm going to bring that email back. So as I mentioned, and I'm happy to say again, the community sampling in this project was really a proof of concept. and this goes along with the RMI project being more open to a broader approach to monitoring offshore wind, the state RMI project. And yes, we're looking, and I'm working to write in more sampling opportunities on these sorts of projects as well as other projects. Community sampling is great for geographically widespread projects. And with the technique that we're using and piloting this study, the community members collect the filter and mail it to us. So they can literally do their work wherever, and we're using a type of filter that's self-preserving. And it can be mailed to us, and then we process from there. So definitely make a connection with Erin, keep in touch. If this project right now doesn't suit what you can do, then I'm working hard to have other projects that include community sampling.
Great, thank you for that. We have some more questions that we'll get to, but a brief reminder right now, Steve's going to help launch the poll. If everybody, while we listen and ask questions, if you can fill out in the next few minutes some feedback on tonight, that'd be much appreciated. A few other questions here. We have Christine was wondering, what kind of filters do you use to directly filter the water? What pore size?
So for the pore size, it's 0.45 micrometers. And that's the same for both the lab and the community filters. The community ones are self-preserving, the lab ones are not.
Great, thanks. And Christine was also wondering, her second question, do sick or injured fish shed more eDNA than healthy fish?
That's a great question. That is, you're thinking like an eDNA scientist in asking that question. I would imagine a fish that's recently been bitten in half by a shark, for instance, would shed a whole lot of eDNA, more than a fish that hasn't been. How that impacts the answer that we find to fish community composition is a tough question. We're trying to get at that by pairing with traditional surveys that sample live fish, like trawl surveys. That's why we need that baseline data.
Right on, and that's we have some really good, astute questions from Ted. He's wondering, related to your last comment there, Jason, are the turbines expected to be built in 2025? Will your baseline be statistically significant in that time frame?
So the answer is yes. I would love to have had 10 years. I'm an ecologist. I think in long time frames. 30 years is that's a time series that we can start to learn something. The BOEM recommendations are for 2 years before, 2 years during, 2 years after monitoring, which is a minimum, and you can, yes, get statistically significant data from that. For other sites, as we're taking the regional approach with the RMI, for other sites that come online, we will have a longer baseline than that. The more we start sampling now. That's a great question. That's the kinds of things that Doug and I and the other people on the team talk about all the time as we're making these plans.
And thanks, Jason. The other part of the question, if you want to attack it, was are the wind farms expected to be started construction in 2025?
Oh, I think Ocean Wind 1 was the only project that was going to be started by then. I don't think there is a project that will be started by 2025 at this point.
Correct. Ørsted's Ocean Wind 1 was going to start construction this year and be commercially operational in 25 or 26, but they canceled that project. They're still holding the lease. The next furthest along for New Jersey's Atlantic Shores Offshore Wind, Project 1 in Cop South. They're expecting to start construction in 2026. And it's about a 12 to 24 month process for construction. Yeah. There are projects nearby that are selling energy to New York. Empire Wind 1 is furthest along. I don't know their exact timeline. John was wondering, can eDNA provide population data as well as species present?
So more often we talk about community data, so the different taxa that make up a community rather than the size of the number of one taxon, which would be a population. I would say that that's not its strength the way that we're using it. There are other approaches. So Emma described the qPCR, the single species approach, where you can measure the relative DNA abundance that relates to the relative fish abundance of a single taxon of fish. In that way, I think that's more of a population level application than what we're doing. We're more interested in community composition.
Right. Thanks,
But
Jason.
it can be used like that. I see a question for Emma.
Yeah, go right ahead. I don't. There's not a prescribed order. Emma, somebody's wondering, how has your experience been working on interning or volunteering, even right on this cutting edge research?
It has definitely, like, since the beginning. I started in July of last year. So I've almost been there a year now, and my experience has been fantastic. I've never had any like days where I'm bored. I'm always doing something interesting. I started out like from the first day, Dr. Adolph was showing me how to do stuff. It was very interesting. And I've been included in every single like part of the project, from going out into the field to processing the samples that we collect. And I always had an interest in science, but this made it even more clear like what I want to do when I'm like in college and what I want to do as a job. So yeah, it's been great. Great people, great experience. It's been fantastic.
Absolutely phenomenal. Fantastic experience for your career trajectory. What year are you in high school?
I'm a senior, so I'm going to be out of high school.
Okay, cool, good. Hopefully we'll get to work together in the future. Jason, I see you're typing a question to Ted. Do you want to continue typing or answer?
So yeah, Ted asked, how do you equate the relative presence of eDNA presence to the actual physical count from a trawl? So I'll post what I typed, but so if you count up all the fish eDNA that we found, and then count up, for instance, the number of striped bass reads that we found, number of fragments of striped bass DNA, that might represent 50% of the total eDNA reads. From the trawl data, we'll look at the total per sample, we'll look at the total biomass of fish caught, and then say how much of that biomass was striped bass. And when you see lines like I showed you, it means that when the relative biomass abundance is high and the eDNA abundance is high, or when they're both low together, you get a positive line through the data. And that's what we'd like to see, because that means that in addition to presence, we can determine that's what I call the relative abundance of taxa within the community. So you can have the same 10 taxa in a yes, it is a percentage comparison. You could have the same 10 taxa in a community, but in one community, it may, the dominant taxa might be sharks, and another taxa, the dominant taxa might be black sea bass. But the names are the same in the two lists.
Cool. Thank you. Very great questions, Ted, and thank you, Jason. Jack was wondering, are you applying eDNA to any monitoring in our estuaries, or do you know of others who do use DNA in estuarine habitats?
I do know of others that use eDNA to monitor estuarine habitats. We had a project in Raritan Bay where we did transects from the railroad bridge near Driscoll Bridge out to the ocean between Queens and Sandy Hook, and we compared trawl capture and eDNA. and found similar results as to what I showed you here. And in fact, we're developing a project. We're recently awarded money from Congressman Pallone's office to use eDNA to look at biodiversity in and around the Raritan estuary, or the Lower Hudson Raritan estuary. So yes, like I said, it's a scalable technique. You basically need the people to go out and sample and send samples back, and it's applicable to open ocean as well as coastal waters and inland waters.
How effective is eDNA at shellfish? You talked about different classes of finfish and marine mammals. Is eDNA effective at shellfish?
So funny you should ask, because that's somebody just asked me that recently in the context of a proposal. And so the primers that we use, like I said, will amplify vertebrate DNA, but there are other primers that amplify invertebrate DNA. What I've seen in the research in terms of scallops, so that's a really important fishery for our area, is that eDNA has been used to detect mass spawning events, where you can take water and monitor it for the high abundance of scallop DNA using a qPCR approach, and get at the timing of mass spawning events. So yes, it's potentially useful in that regard. I don't think that's been looked at as much as finfish have been looked at for eDNA.
Right on. Thanks, Jason. So I think then it could also be applied. John's asking to look at colonization maybe of different species at living shorelines in the estuary. That seems like a reasonable application where it could be effective.
Yeah, so I think that's a good point. Where you have habitats with lots of rugosity and nooks and crannies where fish can hide and you can't easily visually count them or capture them or want to capture them. eDNA in a localized area like that could be, that's the kind of environment where it can be very useful. Yes.
Good. And pitch towards all of you. The last three questions we have are somewhat related. I'll merge them. Have you dealt with some of the experiences of working with on sensitive issues related to offshore wind and community science, given some of the issues that have arisen around marine mammals? Has that impacted your work? And how have you navigated that in working in this space? Sorry, I didn't pitch that very effectively.
Aaron or Emma, I'm curious to hear your thoughts. You're new into this.
So far, I haven't had any super negative interactions. Most of the time, it's just people asking me questions and what my opinions are. And usually my answer is, I don't have an opinion. We'll just see where this goes. But I think the biggest question is, what is the alternative? I think most of us know that we have to steer away from fossil fuels. So what does that look like for us in the future? I don't know, but I think it's a start.
Emma, thoughts?
No, I'd say about the same thing. Anytime Aaron and I go down like the coast to do our sampling, we're always asked by a bunch of fishermen what we're doing. It's never anything like negative in a big aspect, really. So yeah, I think it's science. We're trying to see what's happening and if there is anything happening, what the connections are. And at this point, you can't really come up with any opinion at this point. So
I will add, I've had conversations with my mom about the whales, and my initial answer is always wait for the reports to come out on how it actually died. From what I've seen, it's usually they get hit by boats. So just look into it before making assumptions. That's my spiel.
My answer to that is I'm a scientist. And that means that I form opinions based on data. There's not a whole lot of data out there to support potential impacts of offshore wind on anything. But I'm very happy to be a scientist involved in the collection of that data that will inform the discussion going on, because it's a very important discussion. It's a huge issue. Climate change is a huge issue. It's going to take big moves to address climate change. Offshore wind is one of them. And whenever you do something big in the environment, it can potentially have impacts on other things in the environment. There's no magic bullet solution to climate change. It's going to be a perfect solution without, having an impact on something else. But we need to, I believe, make plans and collect data to be able to scientifically address what those impacts are moving forward, so that we can make informed decisions about different technologies that are being proposed to address climate change. I'm happy to be part of that.
Thank you, team. Appreciate your perspectives on that. We are just past time, but I'm going to honor the 2 questions that we have remaining. One is from Steve. He knows a lot of fish management decisions involve knowing the sex or sex ratios of species. Can eDNA determine that?
So that's a great question, and this is why trials are not going anywhere anytime soon. It's not the kind of thing that eDNA can easily address the way that we're applying it, and the way that most people apply it. The technology is not there yet. So you're absolutely correct. There are lots of things about fish that need to be measured in trawl surveys that tell us about fecundity, age, size class, growth rate. eDNA's strength is measuring biodiversity in communities and presence of different taxa in different areas. That's what it has to contribute. It's not going to replace a lot of the techniques that do those other things for the time being. Great question.
Often used to also track invasive species, right? Or endangered species that are hard to catch.
That's where Fisheries eDNA started tracking the invasive carp. Back, how long ago was that? 15, 20 years ago?
I remember science conferences of seeing talks on this in like 2014 or 15.
Yeah. Just like it takes more than a hammer to build a house, it takes more than one tool to monitor fishes. eDNA is just one of them.
I like your analogies. They're different than I'm used to, so you're adding to mine. Just like kind of a little free ranging there before, like I left this one last question from Dan. What changes do you expect, or what changes are you keeping a close eye on for some of this work for the impacts of wind farms on marine life?
Aaron or Emma, I'll throw this one to you first, if you have a thought about it.
Well, there is the aspect of artificial reefs. So there will be structure there, so that will provide home for species. So we'll see that happening. As in terms of migratory species, I don't know enough to comment on that. I'll pass it along.
Emma?
I do not come from an area of expertise to comment at that level. So, Dr. Ada, I'll pass it back to you.
So I think the idea that structure-associated fish are going to become more dominant within the wind farms is seems reasonable, and that seems like the kind of thing that we could, with eDNA and other techniques, easily detect as we do our before, during, and after monitoring. As an ecologist, I wonder whether or not the change in the abundance of those fish could drive changes in abundance of other prey or predator fish on the organisms that are going to be enhanced just because of habitat availability. So I guess that's my main thought on how things would change would be structure associated fish with some maybe outward expanding impacts on other fish that consume them or that they consume.
Great. Thank you for your perspectives on that. And also what I thought was unique in your work with the community science is to work on this, along the shore in the surf zone. And somebody mentioned the cabling routes, which up in Raritan Bay and some other joint sampling you were doing. I think the sampling in the surf zone would be very unique and effective here for seeing what the cable landings effect might be on fish. So very cool there as well.
Thank you.
Great. So with that, we got through all the questions, just a little bit in overtime for some extra fun. Thank you again, Jason, Aaron, and Emma for joining tonight. A really great presentation and contribution to this series, The Elements of Natural Resource Management. The recording will be posted in a few days on our Earth Day Every Day webpage and our Rutgers Extension YouTube channel, so people can share it with their friends, view it again. The link's in the chat again for registration for the next seminars. This is number 2 of 6 of our spring seminars. The next one is Monday, April 29th, in a few days. Dr. Joe Paulin from Rutgers University talking about black bears in New Jersey. We have a few other marine ones coming up as well later on in May and June on offshore wind energy again, and also humpback whale ecology off the coast. So hopefully we'll see you on a few more of those. The poll is still open. If you're at your last second, fill that in, but we're about to close that and stop the recording soon. Thank you for all the participants. We still have 39 with us. That's impressive. Hope you enjoyed it tonight, and we'll see you again soon. Thanks again, everybody.
Thank you, Doug. Thank you, everybody, for listening.